Hi, my name is Paul Gordon, and I am a bioinformatics support specialist at the Alberta Children's Hospital Research Institute, the next generation sequencing bioinformatics and genomics platform. So what is next generation sequencing would be the first question, and essentially what it is new technology that allows us to do DNA sequencing at a scale that is many magnitudes faster, cheaper, and more reliable than ways that we were doing it before. And what this has done is opened up whole fields of research in terms of medical genetics and answering questions for people who have disease, and also in discovering mechanisms for how biological processes work in model organisms and in humans. So essentially my role is to take the gigabytes and terabytes of data that are generated in the wet lab by these machines and then distill all that information down into a spreadsheet or small amounts of data that can be reported back to clinicians or research scientists to say these are the most likely causes of disease in this patient based on their genetic makeup, or to try to discover what are the genes that are involved if it's in a research setting in a lab where they're doing things in test tubes. So what does this mean to the people on the street? Essentially, what we're getting to is an era of personalized medicine. And personalized medicine, you're not just giving a certain treatment to someone because they have a disease. You're giving it to them because they have a very specific genetic makeup which will affect their reactions to drugs, for example. And also we can open up new questions in terms of trying to answer things that we couldn't tackle before because of the scale of data that was required. And I'll show you a couple of examples here. Say, for example, we had a patient with a particularly nasty viral infection. What we could do is take different samples from that patient over time, if it progressed from tissue to tissue, and then look at the specific genetic changes that are happening within those tissue compartments in order to figure out if this was a normal progression of the disease, if that was just bad because of the particular genetic makeup of the patient, or if something actually changed in the genetics of the virus. It mutated inside the patient. So that's the kind of thing that I do in terms of writing software to do these kinds of analyses. Another type of analysis that I've been working on lately, and these are the kinds of things that we can do with the new sequencing technology that we couldn't do before, is monitoring of organ transplants. So we're working on procedures for trying to figure out if there's rejection of an organ that has been received by someone by looking at how much of the donor DNA is in the blood. If you go above a couple of percent, you probably have rejection, and we can tell this without having to invasively go into the chest cavity and see if there is rejection happening at the microscopic level. And this requires doing some programming and mathematical modeling, and that's really what interested me in this whole thing in the first place, in terms of combining my curiosity about biological processes together with my programming knowledge.